Good afternoon. My name is Brian Horsburgh, and I'm the CEO of Immunova. Immunova has proprietary nanotechnology that we apply to solve problems in immune oncology. Our first application is focused on enabling IL-2 therapy. This slide highlights and summarizes the benefits and features of IMM-01, which are critical to drug development. The IMM-01 target product profile reveals a highly novel, first-in-class IL-2 medicine that retains the ability to selectively expand anti-cancer T cells, but not immunosuppressive Tregs. Our development plan proposes an IND be filed within 12 months of financing that will evaluate our drug in PD-1 refractory patients. In terms of drug supply, we have GMP processes in place with an established CMO. And finally, we have freedom to operate and a robust patent portfolio protecting our technology and our drug assets. So why are we focused on IL-2? It is well known that IL-2 levels correlate with cancer survival, as can be shown here, in that higher levels of IL-2 in the blood result in increased survival and also cures. This led to the development of Proleukin, the first recombinant IL-2 therapy. Proleukin can be curative in patients, with approximately 10% of melanoma patients being cured of their cancer. Our goal is to use nanotechnology to increase the number of cures from IL-2 therapy. Although IL-2 can be curative, it does have a number of drawbacks. The first is it is highly toxic. Large doses of IL-2 are required for efficacy, but this results in cytokine storm and pulmonary edema. This toxicity has hampered Proleukin's use in the clinic. The second drawback is that IL-2 can both activate or suppress the immune system, and we need an IL-2 that is biased towards immune activation. However, natural or endogenous IL-2 promotes immunosuppression and not activation. This leads to a paradox. All exogenous IL-2 variants create a negative feedback loop. What this means is that any IL-2 variant will turn on production of endogenous IL-2, which will expand Tregs, which will suppress the immune response the treatment was meant to activate. In short, increasing Tregs blunts IL-2 efficacy. So let me present some real-world data on Treg immunosuppression. This data has come from a number of companies working on various IL-2 variants, as shown on this slide. So regardless of the IL-2 variant mechanism of action, all of them result in an increase in immunosuppressive Tregs, ranging from 2 to 27-fold. Triggering endogenous IL-2 in patients is a significant problem for IL-2 therapy and efficacy. What this means is that any effective IL-2 therapy will, of course, need to be safe and effective. In brief, that therapy will need to prevent Treg expansion, prevent immune-mediated toxicities, and crucially, retain efficacy. So, manipulating IL-2 signaling has been explored as a strategy to modulate immune responses in the treatment of cancer. IL-2 binds to a receptor comprised of 3 subunits termed alpha, beta, and gamma. Binding to all 3 subunits results in immune suppression and expansion of Tregs. However, binding of only beta and gamma subunits results in expansion of T effector cells. This observation led to the development of a number of so-called non-alpha binding IL-2 variants. However, these non-alpha variants have either failed or are very disappointing in the clinic, but what they did do is highlight yet another IL-2 paradox. So, recent data has shown that alpha signaling is required for expansion of anti-cancer T cells and for PD-1 synergy. This explains why non-alpha IL-2 variants failed in the clinic. The non-alpha variants expanded the wrong T cell subtype. They expanded bystander or anergic T cells, resulting in poor efficacy. This begs the question, if CD25 or alpha signaling is required for Treg expansion and for expansion of anti-cancer T cells, how do you expand the T cell subtype you want without expanding the T cell subtype you don't? To answer this question, we need to understand the interaction of IL-2 with its receptor subunits. This slide shows a cartoon demonstrating that any IL-2 variant that you deliver that binds to the receptors on effector cells will result and turn on production of endogenous IL-2. And endogenous IL-2 can expand TRegs, but only in the presence of another cytokine called TGF-beta. This slide shows that both IL-2 and TGF-beta are required to maintain and expand Tregs. Thus, to answer our question, effective IL-2 therapies need to retain alpha binding and inhibit TGF-beta. But getting an IL-2 alpha and a TGF-beta inhibitor to the right place is difficult and requires a bioengineering solution. And we have that very solution. Our platform technology is a core shell nanoparticle. The shell is a liposome, which is well understood by regulators. What makes it special is our proprietary hydrogel core, which achieves two important features. First, it allows us to solubilize and load drugs with very different chemical properties. The second feature is that we achieve controlled release of the drugs. For brevity, I'll summarize the important features of our IL-2 therapy and show you some data on the following slides. Our nanoparticles are taken up by immune cells and tumor and thus allow delivery of our drug cargo to the right place for the right time. In this case, we can load a hydrophobic inhibitor of TGF-beta and the hydrophilic IL-2 protein. This formulation, IMM01, addresses the IL-2 paradoxes as it retains alpha signaling, but Treg expansion is prevented by blockade of TGF-beta. Nanomedicines bring a number of additional advantages by shifting the therapeutic window. What this means is that we get more drug activity with less drug. So on a molar basis, the efficacy of a nano-formatted drug is increased relative to the native drug. As I'll show you in the next slide, IMM01 is a potent monotherapy and is curative in a number of animal models. The nano-formatted drug is less toxic because you need less of it. Furthermore, the PK and biodistribution is governed by the particle, and this means that less drug goes to lung. And this reduces the likelihood of pulmonary edema, which is the major toxicity for patients. We have now completed IND-enabling studies and are preparing that IND to evaluate IMM01 in PD-1 refractory patients. So let me show you some data. Here we have a survival curve of animals with metastatic disease that have been treated with IMM01 or the checkpoint antibodies CTLA-4, PD-1, or the combination of both. And what you can see very clearly is that 60% of the IMM01 treated animals survive through day 60, where by day 25, all the checkpoint monoclonal antibody treated animals are dead. So there's a significant survival advantage. As expected, if you combine IMM01 with PD-1 or CTLA-4, you actually further enhance those immunotherapies in increasing survival. So here we have 100% of the animals are alive with treatment with IMM01 and both checkpoint monoclonal antibodies. So switching to toxicity assessments, we've evaluated IMM01 in GLP tox studies. At our highest dose, we did not observe any immune-mediated toxicities. As expected, histopathology of the lungs showed no change. This means that our no-effect level dose is our highest dose. IMM01 is highly tolerable and safe in the rat, and our expectation is that it will be safe and well tolerated in patients. So they ask, we require $3 million to make clinical supplies of drug and file the IND. We need approximately another $8 million to conduct the clinical trial and hit a significant value inflection that could represent an exit for shareholders. Some of those proceeds will be used to expand our pipeline, and our next focus will be to significantly increase the drug payload capacity of an antibody conjugated drug by conjugating our nanoparticles loaded with drug, thus increasing the drug payload over 1,000-fold. We believe this approach would be dramatically more efficacious in patients. As mentioned in our opening slide, we have freedom to operate. The technology and pipeline are protected by over 25 granted and pending patents that cover all of the major markets. Our proprietary nanoparticle technology is standalone and doesn't require any third-party licenses. Finally, in order to execute and impact patients, we are assembling a team of individuals who have a track record of delivery. We've just onboarded John Tier, a biopharma veteran with nearly 30 years of CMC and R&D experience. John spent his career at Chiron, Bayer, and Mission Bio. We've identified a CFO and a CMO who could join when we complete our fundraising. Our current advisors include Sandra Panham, who spent her career in investment banking and venture capital and sits on a number of private and public biotech boards. Professor Ruth Plummer of Newcastle University is an internationally recognized oncologist with deep experience in evaluation of novel drugs for phase 1 studies. And finally, Ephraim Fuchs is a co-founder of the company and an internationally recognized immunologist and immune transplant surgeon at Johns Hopkins. The team is focused on delivering new impactful medicines for patients, and we're excited to share this vision with you all. Thank you for your time, and if you have any questions, please ask. Thank you.